Hi, I'm Eric Durand. I'm a computational biologist here at 23andMe. I've been with the company for about 2 years now, and I'm mostly working on their ancestry products, which range from a variety of different problems. Before that, I was a postdoc at UC Berkeley for another 2 years, and even before that, I was a PhD student in France.
We really want to know from a computational biologist is how do you determine IBD or identity by descent?
Yeah, so first maybe we should define exactly what IBD or identity by descent is. So IBD is something that refers to a chunk of continuous DNA, and we say that two individuals share a chunk of DNA identical by descent if this chunk has been inherited through a common ancestor without recombination. It means that you inherited these segments from the common ancestor however far back it was in the past, and it was never broken by a recombination event during meiosis. So if you want the simplest example of identity by descent, maybe is between a parent and his child, where because of the way, if I refer you back to high school biology, because of the way chromosomes are inherited, a child will inherit one chromosome exactly from each of his parents. And so a child is 50% identical by descent with his parent, and in this case a common ancestor is the parent himself, and the child is 50% IBD with the parent.
And so it doesn't matter how big the segment is. The segment can be the whole length of the chromosome itself or anything shorter than that?
Exactly. So if you only look one generation back in time, the segment will cover a full chromosome in the child. The full chromosome in the child will be identical by descent with his parent. If you go one more generation with a child is about 25% IBD with each of his four grandparents, but there already the 25% is only approximate, and it can vary. You can be more IBD, you can share more IBD with one grandparent than with another, just because of the way recombination occurs.
I see. So there may be an average IBD, but it doesn't have to be exact because, like you say, the recombination doesn't happen in the same place every time.
Exactly, and also because of our ability to detect it, there is some error, some statistical error around the estimation of IBD, which in the end will lead to slightly different estimates. But it should be pretty close to 25%. And the further back you go in time and the more variation you have between those percentages, and also the shorter those segments become and the harder they become to detect. So if you want to relate IBD and IBS, for example, the main difficulty with IBD, I guess, is twofold. Is first, we do not observe the recombination process. It would be great if we had some kind of black box or oracle that tells us, well, this is where recombination occurs. But we don't see that. And even worse, we don't even know the genealogy that relates to individuals. Even if they're kind of closely related, if they're second cousin, most people just ignore the tree that connects them. So you don't know how far back in time they were connected, and you don't know where recombination occurs. So the main problem is that you cannot observe IBD directly from the data. You can only observe a proxy of IBD, which is IBS, identity by state. You can definitely detect segments between individuals that are identical or roughly identical. And so this is how an IBD algorithm works. We start by scanning the chromosomes and finding long segments of IBS, and that gives us some kind of candidate pool of IBD segments, and then we apply various filters from these sets to extract the IBD segments. And those filters are varied. They depend on the length of the segment, meaning that it's pretty unlikely to have a very long IBS segment that was not inherited by a common ancestor. We also apply filter for the frequency of the segment that was inherited. If the segment is very, very frequent in a population, maybe it's not IBD, it's just IBS, and that arose to being fixed in the population because maybe of natural selection or other reasons. So frequency, quality of the segment, if it contains many mutations, then it's unlikely to be IBD. We have a lot of filters to explain.
Yeah, it sounds like you really have to think a lot around how to make sure that it's a true identity by descent and not, like you say, either coincidental or something else. So let me make sure I even understand this. The smaller the segment that appears to be commonly shared, the more likely it could be that it's just a match of identity by state by chance with someone random in the population.
That's correct. So maybe I should clarify the definition of IBD a little bit, because under the definition I gave, actually everyone is IBD everywhere, just because we all have a common ancestor if we go far enough back in time. So when we talk about IBD, we usually restrict ourselves to the recent past, say, I don't know, a few tens of generations, maybe 1,000 years. And what's beyond this point, we just don't have the power to decide anymore if it's IBD or not. But if you look small enough, everyone is IBD everywhere.